X-ray, gamma ray, alpha, beta, all these rays, they are ionizing radiations. But talking about the non-ionizing radiations, so basically here you have the UV rays. Okay, this much is clear?
Yes, ma'am.
All right. So talking about the physical mutagens, so we'll say that radiations are the most important physical mutagen and what are mutagens? What are
Factors
mutagens?
that cause mutation?
Yeah, all sorts of agents that cause the mutation. So now talking about this radiation, the radiations are of two types. The first one is ionizing and then there is non-ionizing.
Ma'am,
Yes,
then what is mutant?
mutant.
Yes.
Anything that has had mutation done in it, that is a mutant.
Okay.
Okay.
Okay,
Now talking about the ionizing radiation, the examples are X-rays, gamma rays, alpha rays, beta rays. And talking about the non-ionizing, so the example is UV. Okay. So these are the examples for the physical mutagens. Now see, chemical mutagens, they are basically this. You must remember this one thing, that they are much more injurious as compared to the physical mutagens. Okay, much more toxic. We'll say that the chemical mutagens are much more injurious than the physical mutagens. Now talking about what are the examples, so we'll write nitrous acid, then there is acridines, and then ultimately there are base analogues. These are your following chemical mutagens. Okay, any doubts up till now?
No, ma'am.
Okay, can you people quickly tell me what is the meaning of haploid?
half the number of cells,
Half the number of,
chromosomes.
right. And what is diploid?
2 times the number of chromosomes.
So the normal body cells that we have, they are haploid or diploid?
Diploid.
Right. And which type of cells are in the body are haploid cells?
Germ cells.
Examples?
Sperm and ovum.
Correct. Now let us move on to the polygenic inheritance and let us see what it is. Now, polygenic inheritance is basically, see, all of those traits that are usually controlled by 3 or more than 3 genes. Okay, so like how one gene in our, like one trait in our body or in any way, it is controlled by one gene, right? But here in the case of a polygenic inheritance, one trait is actually controlled by 3 or more than 3 genes. Okay, Like, for example, if we talk about human height, okay, the height in the case of humans. Now, not every single person here is a dwarf, nor every single person is like 6 feet tall. Like, there are so many people in between the two extremes, right? Isn't it?
Yes,
Yes,
ma'am.
ma'am.
Hmm. So here we can say that there's like such a huge range of possible height, right? Same goes with the human skin color. There are so many ranges. Like there is light brown, there is darker brown, there is deeper, even darker brown. So you'll have a whole range of possible skin color also, possible height also, right? So all of these traits that are actually controlled by three or more than three genes, they are known as polygenic traits. Okay. Basically, in this type of inheritance, what happens is that the phenotype, okay, whatever is the phenotype, it is actually because of a combination of each and every allele. So you can say that like however many alleles will be there, they like the effect of it will be additive in nature. Okay, it will be a collective effect of all the alleles present there. Okay, and like for an example, let's just take like for human skin color. Okay, so if capital A, capital B, and C control the skin color in the case of humans. So if we are writing it in capital form, so we can denote it like as they are dominant ones, right? So now let us make the dominant alleles or dominant forms. So capital A, capital B, and capital C, capital C. Okay, now whoever will have this set as a genotype will have the darkest skin tone, isn't it? Because it is the dominant one. So whoever is going to have this type of genotype, then the phenotype will be the darkest skin tone possible. Now, talking about the recessive form of this thing or the recessive allele. Now, can you tell me what will be the skin tone of the people having this type of genotype?
White.
Yeah, meaning the lightest skin tone, right? Like how the dominant ones, like all the dominant this set of genotype had the darkest skin tone, same way the recessive one will have the lightest skin tone. Like these are two extremes, isn't it? So now just imagine there are so many different kinds of skin tone intermediate to these two extreme condition, right? Meaning here all of the alleles are contributing towards the phenotype. So we can say that this is the whole concept of the polygenic inheritance, meaning when three or more than three genes actually affect any phenotype, that type of inheritance is known as polygenic inheritance. Is it clear?
Yes, ma'am.
Yes, ma'am.
Also, we took two examples. The first one was human height, the second one was human skin color. Also, if you want to take some example from the plant side, so the kernel color of the wheat, that also varies and it is also a polygenic inheritance. Okay, so now let us see more about it. So, talking about the language, so you can say that when the traits are controlled by three or more than three or more than three genes are called as you can write it as the phenomenon. The phenomenon when the traits are controlled by three or more than three genes, that phenomenon is called as polygenic inheritance. polygenic inheritance. So is polygenic inheritance clear to you?
Yes, ma'am.
So here, like the effect of all sorts of alleles, what is the effect of it? Hmm?
Changes the phenotype.
Yeah, so you can say that all the alleles, they like the effect of each of the allele present is additive. It gets added and then it contributes to the phenotype. Okay, so the effect of each allele is additive. This is important to remember. Okay, this particular line, it is additive and each allele contributes to the traits of phenotype. So this is your polygenic inheritance. Now, let us talk about another phenomenon, that is pleiotropy. Now see, when one gene can exhibit many different types of phenotypes, okay, multiple phenotypic expression, that type of a gene is known as a pleiotropic gene, and this phenomena is known as pleiotropy. So just imagine one gene that is actually creating or exhibiting multiple phenotypic expressions. And the examples here over, like here, it is skin pigmentation, reduction in the hair, starch synthesis in the pea. All of them are controlled by one gene only. Okay, like each of them, like skin pigmentation controlled by one gene, but so many types of phenotypes are there, right? So this is what pleiotropy is. But before that, let us write down the examples of polygenic inheritance. So here you write down human height. After that, you write down human skin color. Another example is kernel color of wheat. Now, coming on to pleiotropy. The phenomenon when only one gene exhibits or shows multiple phenotypic expression. Okay, when only one gene would exhibit or show multiple phenotypic expression, this phenomenon is known as pleiotropy, and the gene is known as a pleiotropic gene. The gene is called as pleiotropic gene. Let us see what are the examples. Like basically here, see what happens. A great example of pleiotropy is the disease that is known as phenylketonuria. Now, this disease occurs in humans and it is actually caused by when mutation is done in the gene, that gene which is coding for the enzyme that is called as phenylalanine hydroxylase. Okay, now this man like this whole disease, that is phenylketonuria, that if this particular enzyme is absent, then the whole phenotypic expression of this particular mutation in the body can be expressed by mental retardedness, skin pigmentation, and reduction in hair. So just imagine, like, one disease is already being caused, and that is phenylketonuria, and because of that you can also sort of like get all these other phenotypic expressions of that particular disease. You getting my point?
Yes, ma'am.
Hmm. So that is your pleiotropy. So here, if we want to write the example of pleiotropy, so we'll write phenyl, sorry, phenylketonuria. Okay, this is caused in humans, and it is actually occurring, this disease occurs because of mutation. Why? Because the this like whatever gene is responsible for coding for the enzyme phenylalanine hydroxylase. Okay, so now because of the mutation, this enzyme is not synthesized much or not synthesized at all. So then this results in the human being like having the disease phenylketonuria, and in this particular disease, mental retardation, reduction in hair, and skin pigmentation takes place. Got it?
Got it, ma'am.
Is it clear to everyone?
Yes, ma'am.
All right, very good. So now let us write down. So phenylketonuria caused by, always remember that it is first of all caused by mutation only. Okay, so mutation in the gene which codes for the enzyme. Always remember this, the name of the enzyme, because or the because of the lack of which the disease is occurring. So which codes for the enzyme phenylalanine hydroxylase. This is the enzyme. Now the occurrence of this disease, the occurrence of this disease is expressed phenotypically phenotypically by, what are those things? First is mental retardation. Mental, I'm writing. Mental retardation. Also, you people switch on your video so that I can see you. I cannot switch on my video today, but you please do so that I can actually look at you. Come on. Hmm. So even though you people cannot look at me, have this in mind that I am looking at you. So mental retardation, reduction in hair, also skin pigmentation. Okay, these are, so you are you understanding this concept of pleiotropy, that like one gene which is responsible for causing a disease that is known as phenylketonuria, and since that disease is being caused, it is being expressed in the observable way, like how? First of all, mental retardation, then there is reduction in the hair, and it is a significant reduction, not just like normal hair fall, and then also skin pigmentation. So this whole thing is clear. So
Yes,
like,
ma'am.
okay, very good. Now if we talk about the example in the case of humans, then that is phenylketonuria, and if we talk about the example in the case of plants, then you can write down the example as to starch synthesis in the pea. Okay, so that is another example for pleiotropic gene. Is it clear? All right. So now let us move on forward and talk about another this thing topic, that is the whole genetic disorders part of the whole chapter. Okay, so before talking about the certain genetic disorders, there is pedigree analysis. Okay, but see, pedigree analysis, there are so many questions that can come related to this particular analysis. Okay, like how you draw sort of like a tree, like how we used to draw family trees. You remember when we were younger? But this is genetic tree. This is a creepy tree. Okay, family tree was beautiful, cute. But this is a very creepy tree because you are actually talking about mating between relatives, mating between unrelated people, also the disease possibly caused because of that. Okay, so see, this whole topic I need you people to understand in a different way. So I'm not going to do it right now. Okay, first I want to complete the topics that are very easy and very like easy and smooth. Okay, so talking about those, we have Mendelian disorders and we have chromosomal disorders. Okay, so two topics are there. We have, first of all, we have Mendelian disorders. Now see, usually from this, as soon as I saw this, I remember in like when I was in class 12th, I had a viva going on during the board exam, like the board's practical, the final one. So in that I was asked like which one of the following, like they gave me a set of diseases, they said four diseases, and out of those four I had to tell them like which one of them is the Mendelian disorder and which one of them is the chromosomal disorder. So always remember that these disorders and where do they belong to is very important to them. Okay, the origin of those disorders. So talking about the Mendelian disorders. Now, see, Mendelian disorders basically they are actually how they are determined. So it is because of mutation in a single gene. Okay, so talking about the Mendelian disorders, so they are determined by mutation in a single gene. Okay, in a single gene, meaning only one gene. When that gets mutated, and all sorts of diseases which are being caused because of that mutation in just one gene. So those types of diseases, they are known as this thing, Mendelian disorders. And always remember, what happens is that these type of disorders, they are actually transmitted to the offsprings. Okay, they are actually inherited by the offspring. Now, and these disorders can easily be traced with the help of pedigree analysis. Like if you like for example, a, a a child from a family had one possible Mendelian disorder. Now you can actually make that pedigree analysis and see, okay, from where is it being transferred? Okay, from where is this actually being getting to the offspring? Like how did the offspring got affected by this? Okay, so talking about some of the most common Mendelian disorders. So the first one is hemophilia. Many questions get asked on this particular disease, even in NEET. So hemophilia is there. Then there is cystic fibrosis. Have you people seen that movie in which Cole Sprouse is there? I don't know what is that name. Like he had cystic fibrosis in the movie.
5 feet apart.
5 feet apart, right? That whole movie is about this disease only. So hemophilia is there, cystic fibrosis is there, then there is sickle cell anemia. Sickle cell anemia is there. Then the D portion is color blind. You might have heard about this disease also, that people are able to see certain shades and not able to see certain shades. So then there is phenylketonuria that we talked about, the pleiotropic one, that is also a this thing, Mendelian disorder only. So phenylketonuria, and then comes thalassemia, and there are many more, but this much is in the NCRT. And to study in depth is actually, I think, let me see there. Or actually, no, all of them are there. Okay. So now these are the following diseases that come under the Mendelian disorder. Okay. Now remember one thing over here, that a great characteristic of Mendelian disorder is that when it will get inherited by the next generation. Okay, so this disease can be dominant and it can be recessive, meaning this disease can be inherited in a dominant fashion also, as well as in a recessive fashion. But how? Basically, what happens is that when it is inherited in a dominant way, it means that whoever the offspring is, like whichever offspring is getting this disease, will definitely have all the symptoms. Okay, will show all the symptoms, will be severely affected. But the one who has gotten this disease recessively, so they are actually, they don't get the disease, but they become the carriers of the disease. So maybe their generation did not get the effect, but now when they'll reproduce, these carriers, when they will reproduce, so there is a 50-50 possible chance that the offspring will have the disease. Getting my point? So they can be like dominant, like sex linked recessive disorder, sex linked dominant disorders. Okay, and always remember it is related to the sex gene, not the body ones. So either it is related to the X or to the Y. Okay, so talking about the Mendelian disorders, they can be sex linked recessive and they can be sex linked dominant. Got it? All right. Now, after that, let us see which one actually comes in the dominant one and which one comes in the recessive one. So for your ease, I have made a chart about it. I mean, yes. So first we will write the serial number, of course, 1, 2, 3. After that, we write the name of the disorder. Later on, we write whether it is dominant or whether it is recessive. Make the chart like this only, okay? And the last column goes to whether it is autosomal or sex-linked. Sex-linked. What is the meaning of autosomal? Hmm? Autosomal.
So body cells,
Hmm. What is the name for sex cell?
germ cells,
And like how autosome, like autosomal,
somatic cells,
that is for autosomal cell. Germ cell is for sex cells. But there is another name that is allosome. Okay. Allosome is for sex-linked cells or the sex cells or the germ cells, and autosome is for the somatic cells. Now, this much is clear. What are autosomes and what are, so like the disease which will be caused because of the body cells or which one will be caused because of the germ cell. Okay, so this is what we are referring to. Now the first one is hemophilia. Let us start with hemophilia here. So hemophilia is the name of the disorder. It is a recessive type of disease. Okay, and it is X-linked, meaning X chromosome linked, meaning it is a sex-linked disease. Now, the second one is color blindness. This is also recessive. And then it is also X-linked, meaning it is also related with the female or the X chromosome. Okay, it can, like the X chromosome can be from any of the parents. Now there is another disease that is sickle cell anemia. This one is also a recessive, but it is autosomal disease. Okay. Now, the fourth one is phenylketonuria. it is also recessive, and it is also autosomal. Then comes cystic fibrosis. Recessive, also autosomal. Now moving forward, coming on to the sixth one, that is thalassemia. It is also the same case. And autism. Now, one more disease is there, which is like I forgot to mention earlier. Now, this one is an extra. This is actually not given in your book, but you should know. You might have heard about myotonic dystrophy. When maybe you were in class 11th, you might have studied about locomotion and movement. In that particular chapter, you might have heard about myotonic dystrophy. This is a muscular disorder. Okay. Myotonic. Can I see? Why are they using I'm writing on it. Myotonic dystrophy. dystrophy. It is a dominant type. Now, this is the only one that is a dominant type of disease, and it is autosome. So, it is dominant autosome. Okay. So, this is the whole, you can say, the what is it, concise version as to which disease, whether it is dominant, whether it is recessive, and whether it is autosomal or sex linked. Now, if you like have a quick look at it also, then you can know like, okay, which way is it being inherited to the offspring. Got it? Any doubts up till now? Now, don't worry, we'll be discussing about each and every of the disease. The first one to begin with, we'll start with colorblind. Because this is so interesting and easy also. So, color blindness. Now, talking about color blindness. So, first of all, like even if you don't know anything about color blindness, okay, if any question comes to color blindness and you don't know anything about it, but one thing you can say that color blindness is a recessive sex-linked disease. This much you can say, right? Even if you don't know anything about it, and that is this answer will be a whole complete answer in itself. But as to know what happens in this, basically, whoever has color blindness, that person cannot distinguish the red and the green color. So that person will never know what is the color of the grass or how the grasses or the plants look like. Okay, anyone who has color blindness, he or she will never know what red color is because they can't make out that color.
So like what colors do they see instead of those colors?
I think they see gray. Or they don't see the color at all. I don't know. Maybe
So like black and white.
yeah, exactly. Like they can actually see the different colors, but not the red and the green. Okay. So this is what happens. Now, always remember that the gene for the normal vision, okay, like normally it is a dominant gene. Okay, it is not a recessive gene. Now, the gene for the vision, it is actually carried by the X chromosome. Okay, first I'm explaining, then I'll write. Okay, so normal gene, it is actually carried by the X chromosome. Now, in the female, color blindness appears when both the sex chromosome carries the recessive gene. For example, this is the gene for colorblind. Okay, XC, small c. Okay. Now, this XC, if it is present in the case here, and let's draw all the possible cases. These are the possible cases, right? Talking about the males, this is the one and only case that will be there. And talking about the females, these are the two cases, right? So here, basically what happens is that either like the male will have color blindness if they have this particular gene on the X chromosome. There is no other way. If the male has it, then he will be colorblind. But in the case of females, the first case, this one that you're seeing, both the X chromosomes are having the color blindness gene. So mean this female is colorblind. But this one, wherein only one X chromosome is having the colorblind gene, the other is not. So this female is known as a carrier of that disease. This is what it means by recessive type of disease. Okay. Meaning, okay. Great question. I have a great question. Can you tell me, okay, this color blindness is a recessive X-linked disease, right? Why is it called recessive? Or how is it denoting to a recessive behavior? If you've understood the previous classes that I've taught, then you can easily answer this. Why and how is it referring to a recessive type? The answer is right there in front.
Ma'am, I'm not sure, but then when it's dominant, it is surely going to be expressed. But then when it is recessive, there's a chance of it being, there are probability of it being expressed or not expressed. Is that right?
I didn't get that meaning. I didn't understand. Like if it is recessive, then
If it is recessive, then there are chances of it being expressed or not expressed because there is no dominant trait present over here, right?
yeah, you're very close. You're exactly very close to the answer, but you're not giving me the exact answer. The question that the answer that you said right now is the right answer, but not the right terms are used. So think of the right terms and your answer will be right. In which way or in which condition is the recessive trait expressed?
When there is no dominant trait present.
And that type of situation is known as what is that?
Codominance?
Huh?
Codominance?
No.
Segregation?
No. It is one of the two situations, like the condition wherein like it is the condition, only condition needed for a recessive trait to be shown. It is a dash, it is a one word, dash condition. Mm-hmm. What I say? You people will laugh if I say. The condition is homozygous condition. Did you not know it? You did, right? Both of you did. Homozygous condition. Do you remember? Homozygous means this, meaning both of the same alleles will be present, right? Both of the same genotype. So that is a homozygous condition. Here, this one is a homozygous condition, whereas this one is what? The third one, wherein the female is a carrier. If the second one was homozygous, what is the third one?
Heterozygous?
Heterozygous. It is a heterozygous condition. Okay, so this much is clear.
Yes, ma'am.
Okay, great. All right, now moving forward. See, when the female would have both, meaning the homozygous condition will be there, then it will be an affected female. she will be an affected female. But if only one of the gene will be there, then she will be a carrier female. Okay. But if in the case of males we are talking about, so here the defect would appear because of the single recessive gene only. Why? Because the other chromosome is the Y chromosome and there is only one X chromosome. Okay.
So males don't get affected by this
No,
disease
they will.
disorder.
If any male would have the gene, they would directly be affected. Okay. But if the female has only one in a heterozygous condition, then she can be a carrier, not affected by it. But if in the case wherein she's carrying both in a homozygous condition, then she will be affected by it. Okay. And now the type of inheritance which is actually shown by the color blindness is known as criss-cross inheritance. Okay. Now in this type of inheritance, meaning in a criss-cross inheritance, a male transmits his trait to his grandson through the daughter. Getting my point? The daughter is not getting it, but the grandson is getting it. The son of the daughter is getting it to his grandson through the daughter. And this is why, like, the daughter is not directly transmitting it to the son. Rather, the male, the grandfather is transmitting to the grandson through the daughter. Okay, so this is the meaning of criss-cross inheritance. Okay. Also, this happens in the case of like this thing, the females also, but in the females, what happens? Like a female would transmit it to her granddaughter through her son. Okay. And a female would transmit it to her granddaughter through her son. So this is a condition for a crisscross inheritance. Okay. Now, first let's write a little bit about color blindness. So here the first line is color blindness is a sex-linked recessive trait in which the affected person fails to distinguish red and green color. Okay, he just fails to do so. Okay, now let us see what actually can, like, how the trait is inherited. Also, there is another thing about that whoever is the colorblind person, he actually cannot tell the difference between the red or green color. Okay, and it does not mean that they cannot do their normal work, but they have problems such as, like, where would it be? During like traffic lights, they cannot see when to stop, they cannot see when to go, because red and green light, both of them are there, right? They just know how to wait. So so yes, it doesn't mean that they cannot do the rest of the work, but yeah, but they don't know how when to stop during the traffic signal. And again, okay, very bad for me to joke on this. All right. Now see, let us look at how the trait is actually inherited. Now, see, they can ask you questions like this, wherein they would give you situations like a normal woman married a colorblind man. what are the chances of the offspring having color blindness? This type of questions can be coming in the exam. So talking about, let's see one example. So cross A, okay, wherein a normal woman is there. Okay, you people make it. First of all, you make it, I'll write it, but you try to make it on your own, okay? So normal women is there and color blind man. Now you make out how many children would have color blindness. First of all, tell me how many offsprings will be there. Then tell me how many of them will be normal, how many of them will be affected. Because this is the only type of question that can come from here. Done? Okay. Tell me, how many offspring?
Uh, I got four.
Okay. How many, both of you got four? Offsprings? Four?
Yes, ma'am.
Good, that's right. Now tell me what is the situation of each of the offspring? Who is affected and how is it?
I got no one is affected.
No one is affected. But apart from that, do you have any other observation? What does
Two
it look?
female carriers.
Two female carriers.
Two female carriers. For both of you? Okay, and what about the boys? Are they normal or are they abnormal?
Two normal boys.
Two normal boys.
Affected.
Great. Very good. Very good. So a normal woman is XX, right? And a colorblind man, colorblind man is X, C, and Y. Now, talking about the gametes from the normal woman would be X, right? And talking about the gametes from the colorblind or affected male would be these. Now, let us make a cross. So, X, C, and Y, the male gametes, and the X, the female gametes. So, the first offspring will be this. The second offspring will be this. Then this and this. So, two of the girls that were born were carriers of the disease which is known as this thing, colorblindness, and the males were normal, two normal boys. Very good. This is how you made it. Great, awesome. Now, always remember that this was one case. Now, if I say the next cross, okay. So here, if I say that a carrier woman married a colorblind man. Now tell me.
Ma'am, can I say it?
Tell me.
One affected female, one affected male, one carrier female, and one normal male.
Very good.
I got this.
Exactly. Very good. So you people have gotten how color blindness is inherited. Okay. Very good. Very nice. I'm so happy. All right. So this color blindness is done. Do you people have any doubts? Anything from color blindness? All right. Now let us move forward. The next disease number 2, which is hemophilia. Now, hemophilia is which type of disease? Recessive or dominant?
Recessive?
Sex-linked or autosomal?
Sex-linked.
Very good. So, talking about the hemophilia, it is an X-linked recessive trait. Now, we already know if it is recessive, then the normal carrier female, which is heterozygous, it will be transmitted from a normal carrier female to the male, isn't it? Now, in this particular disease, what happens? That there is a blood clotting factor, okay, which is a proteinaceous thing. So, because of that defective formation of that blood clotting factor, so the exposed blood of the affected individual fails to coagulate. So anyone who has hemophilia, so here what will happen is that they will, like, whenever if they get a cut, or if they undergo a surgery or anything, their blood won't stop like bleeding. Like, they won't stop bleeding because the coagulation factor, the blood clotting factor, is defective in them. Okay. So, talking about this disease, what happens is that defective blood clotting factor is produced, because of which the blood of the affected individual, the blood of the affected individual would fail to coagulate. So blood clotting won't be there. So even if like a person would get a very minor simple cut, the bleeding will never stop. Okay, even if it is a normal scratch, like the simple cuts that we get, even sometimes, have you ever experienced that horrific paper cuts, the longer paper cuts? I hate them. Ugh. So just imagine you got a paper, no, somebody has a paper cut and that never stops bleeding. So that is hemophilia. Okay, why? Because here the clotting factor is defective. Okay, now a very interesting thing here is that the possibility of female becoming a hemophilic is extremely rare because, first of all, in order for a female to be affected by hemophilia, the mother of such female has to be a carrier and the father should be hemophilic, right? When a carrier mother is married to a affected father, then this condition would result in a female being affected by hemophilia, right? Now, you must remember that why is the possibility of hemophiliac female very less or rare is because, first of all, the hemophilic father, like this one of the female, first of all, these hemophilic males, they actually become unviable in their later stage of life. So it means they cannot reproduce. Okay. Now, just in case, let's just say that, okay, it did happen. Now, the hemophilic female like was actually conceived. Okay, so that female would be this, like the genotype will be this. Now, hemophilic females, they actually die before the birth. So this is the reason why the possibility of a female becoming hemophilic is very rare. The females can only be carriers of hemophilia. Got it? So we can write here that the possibility of a female becoming hemophilic is very rare is very rare because the mother of the mother of such a female has to be at least a carrier and the father should be hemophile. Okay, and the hemophilic father would or the hemophilic males would be unviable in later stages of life. Unviable. Okay. So another thing is that hemophilic females die before it dies before birth. Okay. So like they can ask you like why is the female becoming a hemophilic very rare? So you know why now. Okay. Now see, thing here when people were actually making pedigree charts or pedigree analysis, if that family pedigree of the Queen Victoria, it shows that a number of hemophilic descendants were there in the family tree or the okay in the pedigree analysis when it was made for Queen Victoria. Now here, what was the reason? Why did the descendants had the this thing hemophilic descendants? So because the Queen Victoria was a carrier of this. Okay, so that is like one fact you should know. So that was about hemophilia. Is it clear? Any doubt? So talking about the hemophilia, so here if a normal man marries a girl who is carrier for hemophilia, then what would happen? A normal man is marrying a girl who is a carrier for hemophilia.
One affected
One affected what?
one
Gender?
affected male,
Hmm.
and then one carrier female,
Hmm.
and then one normal female and one normal male.
Very good. Exactly. That is how it would be done. So hemophilia is clear to you people?
Yes, ma'am.
All right. Very good. Now let us move on to the next disease, that is sickle cell anemia. The third one. Sickle cell anemia. All of these diseases are very unique, to be very honest. Now, let us see how and what sickle cell anemia. Now, see, sickle cell anemia is an autosomal recessive disorder. And now, the easiest way to remember all sorts of Mendelian disorders is like all of them are autosomal recessive, except for the hemophilia and the color blindness. They are recessive, yes, but they are sex linked recessive. Also, myotonic dystrophy is dominant autosome. Got it? All right. Now, talking about the sickle cell anemia, so it is autosomal recessive disease. So, meaning here what will happen that it will be transmitted from the parents to the offsprings when both male and female individuals are carriers for the gene. Okay, meaning a heterozygous condition. So, sickle cell anemia will be inherited in the heterozygous condition. Okay, so in this particular case, like the gene denoting for it is HB. Okay, so HBA and HBA. This is normal and this case is homozygous. Do you know what is HB? Hmm?
Hemoglobin?
Hemoglobin. And this is normal carrier. The sickle cell anemia is denoted by an S. So the hemoglobin that is carrying the S gene and one normal gene, that will be the same. The carrier. But it will be normal because carriers are not affected. They are just carrying the disease. Now comes this. As, as I said, that it is an autosomal disease. And the second one was it is a recessive autosomal. Since it is recessive, it can be shown only in homozygous form, right? Homozygous recessive form. Also, another thing is that since it is autosomal, like how here we were dealing all the disease with X chromosome, right? So now what will happen is that we are dealing with HB, that is hemoglobin, that is not actually a sex chromosome, right? It is the chromosome for blood, right? It is a body cell. So that is how it is, this thing, a autosomal recessive disorder. Now, for all those individuals that do have the sickle cell anemia, so this one, meaning in the homozygous condition, so they are diseased, right? Obviously, they are properly affected. And another thing is that they die before attaining maturity. Okay, so they don't also attain maturity. So these type of individuals that will have the sickle cell anemia, they will die much before attaining the maturity. Got it? Okay. Now see, for all those carrier humans, carrier of sickle cell anemia, so they would have a 50% chances of either transmitting to the offspring or not, isn't it? because they are the carriers for this particular disease. Now, let us talk about what actually happens in this particular disease, which is sickle cell anemia. So here, this whole disease is because of mutation, more particularly transversion. Now, can you tell me what is transversion first? Transversion type of mutation. Transition, transversion, two types of mutations are there, right? Substitution mutation. What was?
Purine with pyrimidine.
A purine is being substituted by a pyrimidine, and that is known as transversion. So this disease is because of the transversion of the gene which is controlling beta chain of hemoglobin. Okay, now see, hemoglobin has a beta chain. Now the gene that controls the hemoglobin for it is getting substituted by another gene. Okay, so here once the gene is mutated, then that gene becomes HBS. This is the mutated gene. Okay, mutated gene. So here what happens? This mutated gene, once it gets mutated, it will change amino acid in the beta chain. Okay, the beta chain is made up of several types of amino acids. Now what will happen? This type, like the HB, the mutated gene, would actually replace the glutamic acid, which is present at the sixth position, by amino acid that is known as valine. So it replaces glutamine or with the valine, and because of this, what happens? Hemoglobin molecule undergoes polymerization. Okay, it starts forming polymers, and because of which the shape changes. Now, do you people know what is the normal shape of an RBC?
Circulars.
But the more appropriate way to describe it is actually biconcave. Okay. Everyone remember. Yeah, I can see your both of yours reaction. Yes, exactly. That's my reaction too. So RBCs are normally biconcave. They sort of look like this, isn't it? Like this, right? So they are biconcave. Now, because of the polymerization from this biconcave shape, they become elongated cells. Now, just imagine something was like a bowl. Okay, something was like a bowl, and then from like a bowl, it became to a starter plate. Okay, the starter serving plate. Now, if I were to ask you which one of them, either of them, will carry more dish in them, whether it is a bowl or the starter plate, what would you say? Which one carries more dish, like more quantity?
The plate?
Huh? How? The plate is not even deep. It's like the starter plates are like just like a tray. Just imagine it like a tray. So would a tray carry abundant? Like you can't carry, like for example, rice or dal or soup in a tray. But can you carry it in a bowl? You getting where I'm coming from? I'm just trying to make it, visually understandable. So when the shape of the RBC changes, so just like how if we switch on like the bowls that we have to serve people, that bowl quickly changes itself to a flat tray-like structure. So then obviously we can't serve soup or something to the guest, right? Same way, when the shape of the RBC changes from this thing, the biconcave to the sickle cell like shape, okay, the elongated shape, then what happens? The oxygen carrying capacity reduces because the RBC is responsible for carrying oxygen. So imagine the soup, imagine the dal or the rice to be oxygen. Okay, so when it was a bowl, meaning a biconcave structure, more and more oxygen was being transmitted all over the body. Now, as soon as it became an elongated sickle cell like structure, meaning a flat or a tray structure, then less oxygen will be transported, and ultimately our whole body would have less amount of oxygen, and we can die also. Why? Because oxygen is so important in the whole entire body. So you've understood sickle cell anemia. And why is this happening? The main cause here is because of mutation. Which type of mutation? So transversion. Now, a purine was substituted with a pyrimidine, or a pyrimidine was substituted with a purine, and because of that, the hemoglobin, the normal hemoglobin, which was this, became this. Okay. Now, this, like, the hemoglobin has a beta chain. Okay, it has a chain like this. Okay, the chain, like any protein chain, a protein is made up of many amino acids. Okay, so now 1, 2, 3, 4, 5, 6, 7. Okay, now at the sixth position, there used to be glutamic acid. It is one amino acid. This is when this type of chain will be there, a normal hemoglobin will be synthesized. Okay, now, or then what happens is that after the normal hemoglobin was made into the mutant type, then at the place of the glutamic acid, valine, which is another amino acid, is being substituted. Then this is being removed and we have valine. Now what will happen? Because of this whole reason, the shape is changing because of polymerization. And ultimately, low oxygen is distributed to the entire body. Okay. So sickle cell anemia is clear to you people? All right. So let us see. Write down.
Ma'am, I have a question. Is that why they ask us to consume gluten-free products? Like, does it have anything with this?
Yeah. See, mutants can come in variety of form. It's not just the rays or chemicals. Maybe the food that has, certain preservatives, certain chemicals, they maybe some of those chemicals can be nitrous acid or nitrous oxide. Some of them can be acridines. You never know. How would you know? Right? And if a person consumes it, maybe this whole thing can happen in the body, right? You never know. So yeah, that is the reason why. So talking the language about it. So this disease is caused by mutation, and also remember which type of mutation. So transversion. And what is, like, transversion comes under which type? Tell me, which type of mutation is transversion?
Gene mutation?
No. But I mean, yes, technically you're right, but not the exact word. The exact word is substitution mutation. Okay. Remember I said, have you people had substitution teachers in your school and blah blah? I said that, you remember? Yes. So you write down, like, near the transversion also substitution mutation. Okay. So this disease is caused by mutation. Which type? So transversion of mutation of what? So of the gene controlling the beta chain of hemoglobin. The beta chain of hemoglobin. The mutated gene, what am I writing? Mutated gene is called HBS. HBS. Okay. Now, because of this mutated gene, the HBS causes one change in the amino acid sequence of the beta chain. Okay, so it actually changes the amino acid sequence. Now, how does it do? So it replaces. What does it replaces and with what? Tell me. Which amino acid is being replaced here?
Glutamic is replaced with valine.
Very good. So it replaces the amino acid known as glutamic acid. In the bracket, you write down GLU. Okay, and it is replacing it by valine, which is represented by VAL. Now, where is it being done? So, at the sixth position. They also ask the position, okay, like which position. So, at the sixth position of the hemoglobin chain or of the beta chain. Okay, now, because of this, the mutant hemoglobin, the mutant hemoglobin undergoes polymerization under low oxygen causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. To elongated sickle-like structure. Okay, this much is clear. Any doubts? Okay, so we have done another. Now coming on to the phenylketonuria. Now see, phenylketonuria is actually sort of like an error of a metabolism, okay, of a metabolism activity. And this is again an autosomal recessive trait. Now, whoever is affected by phenylketonuria, they do not have the enzyme that is known as phenylalanine hydroxylase, and this is a liver enzyme. Okay, and what is the function of it? Is that this particular enzyme actually converts the amino acid phenylalanine into tyrosine. Okay, so it converts phenylalanine into tyrosine, and as a result, what will happen? When phenylalanine is not converted, it starts getting accumulated, isn't it? When something is not being reacted upon. So when it gets accumulated, it gets converted into phenyl pyruvic acid. Okay, and because of that, it starts getting accumulated in the brain and it results in mental retardation. And you know how phenylketonuria is also an example of pleiotropism, right? Okay. Now, let us write down the language for phenylketonuria. And since it is autosomal disease, so usually you just need to know about it and there is no need to actually create the crosses for them. So phenylketonuria. Now let us write about that this is an error of metabolism it is an autosomal recessive disease. Autosomal recessive disease the individual affected by phenylketonuria affected by phenylketonuria. Lacks the enzyme called which is known as phenylalanine hydroxylase. Phenylalanine hydroxylase is the enzyme that gets lacking in the person who has been affected with phenylketonuria. So, let us see now this enzyme. You should know the function of the enzyme also. So, this enzyme converts the amino acid phenylalanine to tyrosine. Okay, tyrosine is another amino acid. So, amino acid tyrosine. So, when this enzyme will not be there, it means that the phenylalanine will not be converted. So, as a result, accumulation of phenylalanine takes place. And it changes or it gets converted into phenylpyruvic acid. From phenylalanine, it gets converted into phenylpyruvic acid. Now you must remember the accumulation of these causes mental retardation. These causes so this is the whole entire disease, that is phenylketonuria. The thing to be remembered here is that it is autosomal recessive disorder. Clear? Any doubts? Also done? Noting it down? All right, very good. Now coming on to the last disease, that is thalassemia. Okay, now see again, this is recessive autosomal genetic disorder, and here it is also caused by mutation. Okay, now see, thalassemias, that they are actually a group of disorders that is caused because of a defect in the RBC polypeptide. Okay, now what happens here, if absence or reduced synthesis of any globin chain of the RBC results in an excess of the other. How? Like free globin chains are there, they are insoluble, and then they start getting accumulated in the red cell, and it causes the death of the red RBC. Okay, so because of that, what happens? It results in the situation that is anemia. Anemia because RBCs are not there, so no, hemoglobin and everything will not be there, and because of that, anemia can be caused. Okay, now see, the RBC, it has two globin chains, alpha globin chain and beta globin chain. And for example, if the thalassemia is being caused because of the alpha globin chain being defective, then that is alpha thalassemia. If it is being caused in the beta chain being defective, then that is beta thalassemia. So the thalassemia is actually of two types, alpha and beta. And another is that delta thalassemia is also there. Okay, so the three types of thalassemia. So this was about thalassemia. What happens here? First of all, that it is a recessive autosomal genetic disorder. So now this is which one? Is it fifth or fourth? Fifth, right? So thalassemia. Talking about thalassemia. So it is an autosomal recessive disorder caused by mutation or deletion. Okay, it is caused by mutation or deletion. Now, what happens here? Defect in the synthesis of globin chain in RBC takes place. Or of RBC takes place. So that is what happened actually. Now, always remember that RBC has two chains, right? One is alpha and one is beta globin. So when alpha globin is affected, that is alpha thalassemia. When beta is affected, that is beta thalassemia. Okay? Meaning here, ultimately what is happening? Abnormal hemoglobin is being formed. Okay? So, talking more about it. So, here, abnormal hemoglobin is formed. This much is clear about thalassemia. Now, what happens here is now again, thalassemia can actually be classified into two types. Okay, thalassemia, we'll have to see the types of thalassemia. So, the first one is alpha thalassemia. The second one is beta thalassemia. And these are the two types of thalassemia. Now, what actually happens here is that production of beta globin chain is affected in the case of beta thalassemia, whereas the production of alpha globin chain is affected in the case of alpha thalassemia. So, production production of beta globin chain is affected. So, this much is clear. Any doubts? Now see, let me see, the NCERT actually they have mentioned which of the chromosome or which of the gene is actually denoting the disease. Now see, the alpha thalassemia is controlled by two closely linked genes, that is HBA1 and HBA2. These are the two genes present on the 16th chromosome or the present on the chromosome number 16. and the chromosome number 16. And it is observed because of the mutation, right? Because of the mutation, what happens is that beta alpha globin chain is not synthesized nicely. Now talking about the beta, it is actually controlled by HBB gene on chromosome 11. On the chromosome 11 controls beta thalassemia. Okay, so this is alpha and beta. Now, uh, see, they can also ask you like how can like both of the in both of the cases, whether it is the case of sickle cell anemia or it is the case of thalassemia, it is about RBC only, right? So, question can arise like what is the difference between thalassemia and sickle cell anemia? So the answer can be that thalassemia is like a quantitative problem where very little globin molecules are synthesized, whereas the sickle cell anemia is a qualitative problem where incorrect globin functioning is happening. Okay, so one disease is a quantitative and one disease is a qualitative. Okay, so let us write the difference between thalassemia and sickle. It is a quantitative problem where too little or too few globin molecules. Quantitative, you understand, right? Incorrect globin chain. You write down incorrectly functioning globin chain. Incorrectly functioning globin chain is synthesized. Okay, so this is actually the difference in between the thalassemia and the sickle cell anemia. Now tell me if you have any problem.
Ma'am, this is the last one.
Yeah, this is last for the Mendelian disorder. Okay, so with this,
Then cystic fibrosis.
yeah, all of those are given as an example, but the in-depth knowledge of that is not given, meaning it's not there in your syllabus. Whereas these 5 diseases, they are important to remember. They're important, like you should know what happens in that, how the disease is transmitted, what is happening, all that thing. You should know about this particular disease. So with this, the thing. Now, Mendelian disorders are over. Now tell me if you have any doubts in any of them. No doubts?
How many diseases do we have in chromosomal disorder?
chromosomal disorder, I think there are 3. You have to learn about 3 only. Okay. Chromosomal disorders are much different from this. Yeah, you have to learn only 3 only. Okay, but before learning about the chromosomal disorders, you must know that, like, see, the chromosomal disorders are because of a mutation in the chromosome. Okay, also which type of mutation? So genomatic mutation. You remember when I was teaching you mutation, I said gene mutation, chromosomal aberration, and genomatic mutations were there. Remember? So before moving on to this thing, I mean, chromosomal disorders, I'll first tell you about certain types of this thing, the mutation which changes, like, occurs in the chromosome. Okay, so talking about those, first you should know about the genomatic mutation. Okay, so talking about the genomatic mutation, so it is change in the chromosome number, change in the chromosome number that brings about visible effects on the phenotype. Okay, because of these, like the number of chromosomes present in any humans, it can actually bring about many effects on the phenotype of the organ. Got it? So there are actually 2 cases under this thing. Okay, the first case of genomatic mutation is aneuploidy. Aneuploidy. The second case is euploidy. These are the 2 cases of genomatic mutation: aneuploidy, euploidy. Now, talking about aneuploidy, it is because, like, any change in the number of chromosomes, it would be different than the multiple of the basic set of chromosomes. Okay, here what happens in the case of aneuploidy, either, like, let us see what are the examples for it. So, trisomy, monosomy, like, for example, usually the case for, okay, let me tell you the types. First is monosomy, then there is trisomy. Okay, these are the 2 most important cases you must know from here. Now, here what happens is that, like, normally we have 1 pair of sex chromosome and we have 23 pairs of somatic chromosomes, right? Meaning the autosome. Now, here, either there can be an extra chromosome or there can be 1 less chromosome. Like, a normal chromosome would be 2n, right? A diploid case, isn't it? Now, if the situation is 2n minus 1, okay, when you subtract 1 of the chromosome, then that case is known as monosomy. Getting my point? And if the case is 2n minus 2, then that is known as nullisomy. These are not in the syllabus, but you should know. These 2 are there. Monosomy and trisomy is there. Okay, so after that, 2n plus 1. Then this is trisomy. Okay, so here either the chromosome number can actually be subtracted, right, reduced, or it can be increased. Okay, here if 2n minus 1 is happening, meaning one of the the number of the chromosome is being subtracted. 2 of the numbers are being subtracted in the case of nullisomy, but here 1 of the chromosomes is being added. Okay, extra set is being added. Getting my point?
So, ma'am, would it be 45, 44, and 47 like that?
Yes, Yes. Yes. Okay. Like, no, this will be the normal case. How much it is? It is 46, isn't it? Yeah. Okay. Yes. Yes. Okay. You were talking about these three, right? So, yeah. Yes. So, the cases that can come under aneuploidy is, we can say that this is the change in the number of chromosomes when either it is added or subtracted. This much is clear? Any doubts? All right. Now talking about euploidy. So euploidy, the example here, basically what happens that when a set of chromosome varies, then that is euploidy. And the cases of euploidy are haploidy, meaning only one set of chromosome is there, and another case is polyploidy, meaning more than two sets of chromosomes are there. So haploidy, only one set of chromosomes, and in the case of polyploidy, more than two sets of chromosomes. But what about if two sets of chromosomes are there? That condition is known as,
Diploid.
yes, diploid. Diploid. All right. So this much is clear. Like this genomatic mutation, two types, and what are the cases? That's clear.
Yes, ma'am.
All right. Now see, if we talk about this thing, the trisomy. Okay. So if I am taking the cases of trisomy, so because of the trisomy of certain chromosome, two types of diseases takes place. Down syndrome, and then there is Klinefelter syndrome. Okay, Down syndrome and Klinefelter syndrome. And because of monosomy, Turner syndrome takes place. Okay, so let's talk about chromosomal disorders now. This mutation is clear to you, right? Because to learn about the chromosomal disorder, we must know what is actually like how is the chromosome number hampered. Now the heading should be chromosomal disorders. Now these disorders are caused due to imbalance in the chromosome number. The chromosome number. So the first case or the first example of a chromosomal disorder is the very famous Down syndrome. Now, this disorder, it was first described by a person whose name was Langdon Down. Okay, so because he was the one to describe, so this disease was named after him only. Okay, so it was first described by Langdon Down in the year 1866. Now, here what happens is that this the disorder develops due to the trisomy of chromosome number 21. Okay, so this disease occurs due to trisomy. This is important to remember. So, of chromosome number 21. Okay, so when chromosome number 21 is actually undergoing a trisomy. What is trisomy? 2n plus 1, right? Meaning one extra chromosome is there. So this situation is 45 plus XY. Okay, meaning 45 plus XY. You know, getting my point? So in total, how many are there? 45, 46, 47. So total 47 chromosome. One extra, then 46, which is the normal number. Okay, so this is the case. This is the situation. Is it clear? Also, now let us see what are the various this thing. What are the various ways by which you can actually characterize as to how the person has a Down syndrome? What is the visible phenotypic expressions that we can see, that we can say that, okay, this person has Down syndrome? Now, a person with Down syndrome shows, you can say, it is characterized by the first one, the person would have the short stature. Okay, short-heighted person would be there. Then the second is small, round head. Small, round head. Apart from that, furrowed tongue. Furrowed tongue. Then broad palm. Broad palm with palm trees. A lot of palm trees would be there. Okay. Then another way by which you can characterize is big wrinkled tongue. Big wrinkled tongue. Okay, and then there are many physical, psychomotor, okay, physical, psychomotor, and mental development. These developments are retarded. Basically, in the physical, you'd have, what are the physical retardedness? So underdeveloped gonads and genitals. And physical, psychomotor, and mental development is retarded in this case. Okay, so these are the characteristics of the Down syndrome. All right, and it occurs due to the trisomy of the chromosome number 21. The number of the chromosome is very important to remember. Okay, any doubts? So this was the first disorder that comes under the chromosomal disorder. Now two more are left, and we will do this in the next class because the time is almost over. So we'll be doing the two more chromosomal disorder. Along with that, we will also talk about the pedigree analysis. And then hopefully in the next class, this chapter will be completed. And then we'll move on to the last chapter, that is evolution. Okie dokie. Now tell me if you have any doubts or anything. Anything that you people want to say, anything that you people want to ask, do so. We have 2 minutes.
Ma'am, for the sickle cell anemia, will they ask us to do a pedigree?
No. Maybe yes. A pedigree, yes. A cross, no. The pedigree analysis may be asked, but they ask it in a very vague fashion. We'll do all sorts of